It's a pleasure to be able to introduce our speaker. So Dr. Goldstein is a professor of psychiatry and medicine at Harvard Medical School. She's also the founder and executive director of the Innovation Center on Sex Differences in Medicine at Massachusetts General Hospital and the Helen T. Moschner Endowed. How is that again?
Helen T. Moschner.
Moschner, thank you very much. Endowed Research Institute Chair in Women's Health. She is a clinical neuroscientist and expert in understanding sex differences in health and diseases associated with central nervous system. Her science is focused on prenatal and early life exposures and risk for sex differences in the co-occurrence of these disorders across the life course, such as major depression, cardiovascular disease, and Alzheimer's disease. Dr. Goldstein has received numerous awards for her work, served on scientific review boards, and participated in strategic planning for NIH Office of Research on Women's Health, and the Institute of Medicine. In terms of her background, she has a bachelor's in psychological anthropology from Brown University, MPH, master's in public health, mental health evaluation from Columbia University School of Public Health, another master's in philosophy from Columbia University, and her PhD in epidemiology and psychiatry from Columbia University. And she also has training in neuroscience from Harvard Medical School, and Beth Israel and Massachusetts General Hospitals, and also a master's, an honorary Master of Arts from Harvard University. So without further ado, I'll turn it over to today's speaker. Welcome, and thank you very much for your time.
Just so I have a sense of who's in the audience, are they people with neuroscience background? Is it your center? Are they students? Is it faculty? A little mix of both? Just so I get a sense of, usually it's so hard to give these Zoom talks. I love to see people, kind of get a sense of so the targeting of this talk. And I just want to get a little bit of sense before I get going.
We're a pretty diverse group. I'd say that a quarter to maybe a third have, they have somewhat of a background in neuroscience or know about it, but we have a lot of people from psychiatry, neurology, and also psychology. And that's just the tip of the iceberg. I mean, we have people from social work, and so it's a diverse crowd.
Great, wonderful. That's perfect, because I'm going to talk a lot about cross disorders. So this is absolutely perfect. And are most people working in aging with your amazing center, or no, sort of across the board, lots of different things?
I think it's across the board.
Okay, very good. Well, let me get started. And the last question, sort of housekeeping question, is we have an hour, correct? Including questions?
Yes. Mm-hmm.
That's good. Okay. So when I was originally asked to give a talk, you all asked if I would tell you something about sex differences in the brain and why we should be thinking about that in medicine in general, and why should we think about incorporating this into the work that we do. And hopefully there is going to be time, lots of time for questions and interactions afterwards, so that we can, I can hear a little bit about what you're doing, and hopefully you will see how relevant this is. I will quickly say I really have no conflicts of interest with this talk, although I am on the Scientific Advisory Board for CalaHealth, which is a neuromodulation company with some equity interest, but there is no, I'll mention a little bit about some of the therapeutics we're developing at the very end, but that's really not the nature of the talk. Okay, sex differences in the brain are pervasive. Boys have higher risk of autism, men have higher risk of schizophrenia, men have higher risk of Parkinson's disease, men have higher risk of almost all of the substance use disorders, alcohol, drugs, gambling, all except for eating disorders. Women have higher rates of major depression, almost twofold, and anxiety disorders. They also have almost higher rates of almost all autoimmune disorders, like multiple sclerosis and other autoimmune disorders. And women also have a higher frequency of Alzheimer's disease. And we now know it is not only because women live longer. So lest you think this is just about the brain, of course, sex differences really are pervasive across almost every chronic disease we know. Cardiovascular disease, for example, used to be thought of as only a man's disease, but it's the #1 killer of women in the United States and in most middle-income countries. The rates of cardiovascular disease differ over the lifespan, the onset, the course, the severity. Diabetes rates differ. Rates even differ within women, very high rates in Hispanic women and in African American women in particular for diabetes. As we talked about Alzheimer's, when we talked about autoimmune disorders. So sex differences are really pervasive in chronic diseases of the brain and the body, but medicine has really been built on models of men's health and primarily male animals. And so my goal and my career over the last 30 years has really been to try to change that. And I feel that we are in a place now where this is really being talked about and thought about and incorporated into our research and our clinical practice in a more intensive way than it has in the past. So I'm very hopeful that this will be part of precision medicine. Now, as I said, we are particularly interested in what's termed multimorbidity or comorbidity. And two or three of the major disorders in which this is really important is depression, cardiovascular disease, and Alzheimer's disease. Now, depression and heart disease are really the primary causes of disability worldwide. That is prior to COVID and will be post-COVID as well, as we talked about some of the sex differences in that as well. The co-occurrence of depression and cardiovascular disease was slated to be the #1 cause of disability worldwide this year. Over 20% of the population, this was all prior to COVID, of course, and women are at twice the risk for that comorbidity. And people who have cardiovascular disease that also have depression have a 3- to 5-fold risk of death from cardiovascular disease. So it is more severely and disproportionately affecting women. And we believe that one of the reasons for higher rates of death from heart disease, aside from the fact that women's vasculature in cardiovascular disease is different, it's a microvascular disease, but one of the other reasons for the high rate of death is unrecognized and untreated major depression. And depression and cardiovascular disease are independent risk factors for Alzheimer's disease, memory decline, and all independent of smoking and lots of other potential other factors, independent risk factors. And this multimorbidity is a major public health challenge because in medicine, we really grow up in silos. We have psychiatry and you have neurology, even the same organ hardly get together in terms of thinking about comorbidities. But certainly the heart, the liver, the kidney, the gut, we usually, think about these things in silos. So there are no effective treatments that are really targeted to multimorbidity. And this is a major public health challenge. And we believe that one way to think about this is actually to think about the sex differences that are shared across these three disorders. And I'm going to make that argument to you today. And so we've been tackling really understanding shared causes of these disorders through understanding the shared sex differences, the causes of the sex differences that cross these disorders, whose origins actually begin in fetal development. And we've been studying this through studying the brain circuitry that regulates how we respond to stress. And as all of you know, stress affects every chronic disease we know. So how have we broken that down to try to look at some of the shared causes of that in terms of brain and physiology? So before I get started, there are windows of opportunity for the study of sex differences, and they really are across the lifespan. This is a lifespan perspective. And that is when the brain and the body are either differentially flooded with gonadal hormones in fetal development and puberty for women in pregnancy, or depleted of gonadal hormones in perimenopause and menopause. And we can use these naturalistic opportunities when sex differences emerge to actually provide some insights into why we see sex differences across these chronic diseases and the timing of the causes of those. And again, this lifespan perspective, even for the aging of the brain. I am a developmental neuroscientist, but so how did I get into aging of the brain? About 10 years ago, I got into aging of the brain, my lab did, because I also discovered that aging of the brain is also about development. It's also about development of the brain. And so I'm going to also make that case to you in a sex-dependent way. So what are some of those developmental risk factors for depression, for example? They're small for gestational age, low birth weight, obstetric complications like preeclampsia, oxygen deprivation. The 1954 flu epidemic showed that in the offspring, a significantly higher relative risk for major depression in the offspring, and that was also true for the 1954 famine. And one of the final common pathways that's been discussed is the disruption of the hypothalamic pituitary adrenal axis in development, or what has been termed prenatal stress models of understanding the risk for major depression. Now, in the early 80s, independent of all of this, in psychiatry, David Barker was studying some of the same things in hypertension, and he was looking at small for gestational age, low birth weight, obstetric complications like preeclampsia, and models of oxygen deprivation, and even the Dutch famine study, which also showed significantly higher rates of hypertension in the offspring. And he independently discussed how the final common pathway for understanding this may be the disruption of the HPA circuitry in development, or again, what he termed prenatal stress models of chronic of cardiovascular disease. And finally, a recent study that was in the mere country of Denmark. Of course, you never, in Scandinavia, you only have ends of countries. But in Denmark, they looked at obstetric complications. They looked at preeclampsia, and they found a significantly higher risk in these women for Alzheimer's disease and even more so vascular dementia. But the vascular dementia actually did not account for the Alzheimer's disease risk. And so again, some of the same risk factors occurring in fetal development having lifelong effects on the brain and the heart. Okay, what about timing? Whenever you give a lecture on sex differences, you really need to talk about timing, because timing matters when you're talking about a sex effect. And the sex effects actually differ across our lifespan, but even within gestation. So for example, if you took some population level studies, which my colleague and I did, and you separated them into trimesters, when were these obstetric complications producing sex differences? We found that not that many in terms of effects that were in first trimester, although there were some, and in particular schizophrenia and autism with a male higher incidence. But most of the sex differences emerged when insults were being produced, or when offspring were being exposed during second and third trimester. And that really was across the board in terms of inflammatory models or metabolic models or glucocorticoid responses. So what's going on in second trimester? Well, in second trimester, what's going on is what's called the organizational effects of gonadal steroids on fetal brain development. At mid-gestation, the beginning of second trimester, the testes begin to secrete testosterone, and testosterone in the male human and the monkey, as opposed to the rat, which I'll tell you about in a moment, but testosterone has direct effects on masculinizing the male brain, as well as indirect effects through its conversion into estradiol. So estradiol also masculinizes the male brain in fetal development, but for humans and monkeys, testosterone has direct effects on masculinizing the male brain. In the rat, actually, it's all estradiol. Estradiol in the rat masculinizes the male rat brain. So it's not the best model. We've used it only for, over 60 years. But anyway, so these gonadal steroids are really powerful neuromodulators, and they affect development of neuronal growth and development and apoptosis, death, every level at neurogenesis right up through apoptosis. So they can get right into the nucleus of the cell and change gene transcription, or they can do fast-acting work at the cellular membrane and do things like release neurotransmitters. So the primary driver of the sexual differentiation of the brain is gonadal steroids. that does not mean there are no direct effects of genes on sexual differentiation. About 15 to 20 years ago, it was discovered that there are direct effects of genes on sexual differentiation of the brain. Art Arnold out in UCLA has really been a whole champion and the father of this. But still, the primary driver are gonadal hormone regulation of brain development. And what's been really interesting is that these organizational effects of hormones really kind of get set in development. Now, I'm not a deterministic person. I don't think that, once you're born, that's it. There are many variations on the theme. There is more variability within sex than there are between the sexes. But you cannot castrate an animal later on and produce the same effects that you can in development. So these organizational effects, some of them get retained throughout the lifespan. Now, the reason I'm saying this is this is really key to our hypotheses about why you can have prenatal influences affecting the aging of the brain 50 and 60 years later. Okay, so many moons ago, over 20 years ago, there's a lot more work that's done out there now, but we asked this question many moons ago. I'm a brain imager, so I'm going to show you a lot of brains. And we asked the question many years ago because it actually hadn't been done in the human brain. Just in gross brain volumes, could we pick up sexual differentiation across the brain? And we hypothesized that you would see greater sexual differentiation in gross volumes in brain regions that were dense in gonadal hormones, which is exactly what we found in this particular study. So just again, suggesting these were 40-year-old adults, 42-year adults, male and female adults. And what's suggested here were that there were things that were retained into adulthood that we could look at what some of those fetal origins were. So the structure actually changes, but not all over the brain, and these structures actually have implications for function. So the most highly sexually dimorphic brain regions in the brain regulate stress and emotion. They also regulate memory, and they also regulate reward. But we're going to talk about stress, emotion, and memory today, and the heart. Okay, so what are some of these brain regions? The paraventricular nucleus is the key relay station in the brain. So the stress circuitry, really the relay station of the stress circuitry, which is really, really important, and the HPA circuitry, is the paraventricular nucleus in the hypothalamus. And you also get arousal in the periaqueductal gray and the locus coeruleus, and in the amygdala, particularly with emotional content of material, of course, in the stress response. And then you have inhibitory control by the hippocampus onto the paraventricular nucleus, the anterior cingulate, the medial prefrontal, the orbital prefrontal. And so the brain acts, there's an orchestrated concert of arousal and inhibiting arousal when we're under stress. And these brain regions are highly sexually dimorphic. They've been implicated as abnormal both in depression and in anxiety disorder. They also are really key to the regulation of autonomic nervous system function. And so they do so by connecting with to the nucleus of the solitary tract in the brainstem, which connects to the vagus nerve, which is the direct electrical conduit from the brain to the heart, which regulates cardiac tone. So you're under stress and your sympathetic goes up and your parasympathetic has to get on board, and you need to inhibit the stress response. So I need to have a lot of parasympathetic control right now so that I can give a talk and not get nervous and blah, 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 so that I can calm down. So parasympathetic regulation of the heart is really important, and a deficit in parasympathetic regulation of the heart has been found to be a major risk factor for cardiovascular disease itself. And premenopausal women actually have a leg up in parasympathetic regulation of the heart, which gets lost with menopause. And parasympathetic regulation of the heart is abnormal in major depression and in anxiety disorders, and actually, as I said, in cardiovascular disease and even Alzheimer's disease. Okay, so now we have this orchestration of brain regions that regulate stress, regulate mood, regulate anxiety. They're highly sexually dimorphic, and they regulate the heart. What else do they regulate? I mean, obviously, HPA circuitry regulates so many things. They also regulate memory. So the paraventricular nucleus is very important in memory function and abnormal in Alzheimer's disease. The hippocampus, the cingulate, emotional memory with amygdala. So there's a big crossover, and not surprising when you're under stress, you have memory concentration issues. And so this is not, or if you're depressed, you've got memory concentration. This is not surprising that the brain is actually involved and shared across mood and memory and regulation of the heart. What else is this circuitry regulating? It's also regulating other physiology aside from the autonomic nervous system. So of course, there's the HPA circuitry that regulates the release of what we term stress hormones like cortisol and the anti-glucocorticoid effect, which is a weak antigen of dihydroepiandrosterone. You've got the paraventricular nucleus. That is the densest nucleus in the brain with corticotropin-releasing hormone that acts on the pituitary to produce adrenocorticotropin hormone that then acts on the adrenal gland to produce these, what we say, stress hormones. And you've got the hippocampus then with a regulation, the negative feedback onto the paraventricular. So again, brain regions regulating HPA physiology. They, of course, also regulate hypothalamic pituitary gonadal, right? The estradiol. When I say estradiol, estrogen, by the way, is a plural word. Estradiol is the primary form of estrogen that drives the brain. There's estrone, there's estriol, but estradiol is the primary estrogen that we talk about when we're talking about brain. Okay, and finally, the stress circuitry also is tied in with our immune system. Not surprising, when we're under a lot of stress, of course, we really have a low immune response. And it turns out tumor necrosis alpha, 1 beta, and IL-6, interleukin 1 beta, and IL-6 are the primary co-activators of HPA response. What does that mean? Well, it turns out they are densest in the paraventricular nucleus, the hypothalamus, and the hippocampus and the pituitary. So there's a real crosstalk between glucocorticoid receptors, gonadal hormone receptors, and immune receptors, all sitting in these really key brain regions that are regulating stress, mood, memory, and the heart. Okay, so the immune system is really important in this. And finally, the vagus nerve is also really important, the regulation of the immune system. So Kevin Tracy beautifully mapped this out and calls the vagus nerve reflex the anti-inflammatory reflex, because the vagus nerve wanders. It's the vagus, right? So it wanders all over the body. It's not just connected to the heart, of course, it's connected to lots of other organs, but one of them is the spleen, and it regulates the release or the inhibition of TNF alpha and 1 beta, as Kevin really mapped out through acetylcholine receptors. And so the vagus nerve is also really important in the inflammatory response, in our stress response, and our glucocorticoid response. So it's a really important, which is, how I ended up getting involved in this neuromodulation world, which we can talk about at the end. Okay, and TNF alpha 1 beta and IL-6 have been associated with major depression, anxiety disorders, cardiometabolic disease, and has been mentioned in Alzheimer's disease as well. All right, so the model that we have been working on is we are working on these prenatal stress, we call them prenatal stress immune models of sex differences in these chronic diseases that onset later on in life. And our model has been to use high-risk pregnancies like preeclampsia and conditions that produce fetal growth restriction. And those conditions have been found to increase the maternal immune response. So she mounts a glucocorticoid response. So she's dysregulated her HPA circuitry. And we've hypothesized, and actually it's shown in some animal work and some of the work I'm going to show you in a moment, that when this occurs during the 2nd and 3rd trimester, it actually has sex-dependent effects on that stress circuitry in the offspring, some of which gets retained along with obviously the genetic vulnerability for these disorders. In tandem with genetic vulnerability, you see a dysregulation of that circuitry we just talked about. You see a dysregulation of the physiology, and that is associated with the onset, sex differences in the onset of these disorders. And my colleagues Stu Tobit and Bob Honda out at Colorado State University have been, they're basic scientists, and we do lots of translational work together. And about 15 years ago, we hypothesized, and have some beautiful papers out there if people are interested in this, how glucocorticoids actually disrupt the gonadal hormone receptors that then disrupt nerve growth factors like brain-derived nerve growth factor, insulin growth factor. And we've been looking at this in relation to the GABA-glutamate balance, which we have found impacted the development of neurons in a sex-dependent way, particularly with females, when they have these glucocorticoid exposures during this critical period of development into the paraventricular nucleus and the hippocampus and the prefrontal cortex. And finally, just serendipitously, although this is not the topic of this talk, Stewart found when he knocked out the GABA B receptor and really looking at the development of the vasculature itself was dysregulated in the paraventricular nucleus, particularly affecting the female vasculature in the PVN. And this was very interesting because the most highly vascularized brain region in the brain is the PVN, which is why I got obsessed with this region. Now, I am a clinical neuroscientist, and so we work with people sort of doing all the controls that you can do with animals. So how do we look at fetal programming of these disorders in people over the lifespan? And I have just been extremely lucky in my career to have had access and availability to a cohort that was established in 1959 to 66 called the National Collaborative Perinatal Project. That was a study of 66,000 pregnancies in 12 cities across the U.S. Unfortunately for us in New England, there were two sites in New England, one at the Brigham and Women's Hospital where I used to be, and one at Brown University. And so my colleagues at Brown, Steve Buca heading this up, and our colleagues in Boston, we started following the adult offspring about 28 years ago. And the New England cohort has almost 18,000 pregnancies. So at this point, we followed several thousand of these people. And unfortunately for us, in the 50s and 60s, NIH had the good sense to collect blood during the pregnancy. And so we were able to also assay maternal sera that's stored at NIH and look at these early immune and hormonal biomarkers. The kids were followed for 7 years. The data is actually public data. It's just amazing. It's an amazing data set. And we have capitalized on it by starting to follow these offspring way back when they were in their 30s, and they're now getting into their 60s. And this is sort of how I ended up getting into sex differences in aging of the brain, because they were going into menopause, and I thought, oh my God, we have to study this in real time, prospectively. And so we re-recruit these adult offspring. We clinically and cognitively assess them. We do, on a subsample, we do structural and functional imaging. Obviously, we're really interested in physiology. We do a lot of hormonal assessments. We do cardiometabolic exams. And now, of course, with all the omics, we are separating out cells. We're very interested in the immune pathways. And we're really interested in what actually gets transmitted that is retained in these offspring across the lifespan. And our idea about this is what gets retained is a kind of hypersensitivity to stress and a hyperimmune system that gets retained across the lifespan, and as if you live in adverse environments, will get enhanced along with the expression of genes for these disorders. Or in the opposite way, if you're in enriched environments, these things can be attenuated or ameliorated. But we think that it has to do with this potentially innate immunity change that happens early in development. And that is something we're looking at. We collect monocyte cells. We're looking at gene expression in immune pathways. And we're now doing, I have a wonderful postdoc who's about to come on faculty, And she's very interested in brain metabolism and the impact of menopause on brain metabolism. So we're also taking these monocyte cells and looking at mitochondrial function and respiration ATP production. So we are quite excited about how these cohorts can really inform us in really unique ways, because these people are the naturalistic, sort of form of these illnesses. Many of them are not patients. These were unselected for either psychiatric or medical status. So we're really looking at, across the lifespan, the kind of natural history of these illnesses. And many of them we see unmedicated, even up to 60% of people with major depression, recurrent major depression, do not get into treatment. This is a chronic and very debilitating disorder. And so we see some of these people who have not even been in treatment, unfortunately. Okay, so I'm going to show you a little bit of data now, just to kind of say, well, it's good to kind of, talk about this, but, show me some evidence, So I'm going to whip through just a little bit of data. We have so many other things that we do with this, and I'm happy to talk about it, but I just want to give you a little flavor of how we attack these problems and what we have done. So I'm just going to show you a little bit of data from this study that we just finished from the offspring at ages 47 to 54, who we are now following again up to age 65. We just launched a new follow-up of these 212 people to look at change in memory and risk for AD with PET imaging. But in this case, these were discordant sibling pairs. So you could do all sorts of kind of cool designs. We looked at discordant sibling pairs, one of whom had preeclampsia, or the offspring had experienced fetal growth restriction, and then an unexposed discordant sibling. And this is a really nice design because you can control for at least within family factors. And so there were about 212 of these offspring. And we assayed the maternal sera for, our key HPA co-activators were IL-6, 1 beta, and TNF alpha. Those were the ones. We also look at the anti-inflammatory effect of IL-10. We've been very interested in the mediating effects of major depression at age 45, but we're really looking at what is the implications on associative and verbal memory. We do structural and functional imaging, and as I said, in this new study, we're actually doing PET imaging. Okay, so I have to show you some brain imaging. Those of you who know about structural and functional imaging, we do, my group is our experts in MR techniques, and so I'm just going to show you some data from that. We look at memory tasks, of course, cognition. We always look at cognition, and we've looked at a couple of episodic memory tasks that Doreen Rentz, who was a consultant with us, who was an expert in cognition in Alzheimer's disease, said that these two were particularly sensitive to memory decline in early midlife. So we look at this face name associative task, which is kind of it's a task that gets at a sort of short-term memory kind of task, for those of you who know this. And there's a selective reminding task called the Bush key. We do structural, as I said, MR. I'm going to show you a little bit of data from the functional MR, where we did a verbal encoding test and an MBAC working memory test. So the MBAC, I don't know whether you guys know this. I'm happy to go through what these are. Some of them are short-term memory. Some are getting at encoding and retrieval. So they're just different aspects of the memory system. I'm happy to talk about those in more detail later. But we look at bold signal. You get in the scanner, you do a task like this, and you need a stimulation task, you need a control task, and you really look at what the brain does under a stimulation task versus a control task. And this is really looking at blood flow. Venous blood flow has paramagnetic properties, so you can pick this up with techniques in an MR scanner. And blood flow, this venous blood flow has been coupled with neuronal activity. So we think about it as neuronal activity, but it really is blood flow with all the issues around that. So we look at blood oxygen level dependent signals, and then we can look at not just one region or a set of regions, but how they talk to each other, of course, because these memory functions are really driven by systems, not by one brain region, obviously. And then we also look at physiology. We collect blood for hormone physiology. We look at heart rate variability for cardiac tone, to look at the components of the RR interval, and we look at immune physiology. And again, we're looking at prenatal immune biomarkers, which were obtained from NIH at the beginning of third trimester. And this is just a little flavor of a functional imaging task from the working memory task, which is a well-known kind of simple working memory, short-term memory task, basically. And in this case, what we really wanted to look at was what was the impact on menopause on short-term memory. So let me just remind you, these are 47 to 54-year-olds. Their memories were intact, right? Their memories were intact, but some of them had gone through menopause and some hadn't. And so what I love about this is that we're not really looking in early midlife for like a major, memory decline. This wasn't a study of mild cognitive impairment. It was how does the brain act under different hormonal conditions as the brain changes in women through menopause, right? Even if they can maintain intact memory function, the brain still may be changing, and we need to understand how it's changing in order to understand later how it goes awry. And also in order to try to target people early for enhancing intact memory function. That's one of the main things we're really interested in. We're interested in this period of time, which the field of Alzheimer's is interested in now, because that's where you need to treat. This is where you need to treat. Once the symptoms start onsetting, we really don't have effective treatments. So we really want to look in the preclinical period. So this is the period we really want to study. And it's really important, this period, to study sex differences. So the hormonal regulation of the circuitry is substantial. And what we found here, just in this working memory, is actually postmenopausal women. We actually find in premenopausal women and in perimenopausal women, actually there is a lower dorsolateral prefrontal activity to get to superior working memory function, which is really interesting. People have looked at this in young women as well, and really sort of defined it as a kind of neural efficiency. And in our postmenopausal women, we're seeing higher activity. That's true for the men and also true in comparison to the pre and peri. So what we've interpreted this as is that the brain starts to get a little inefficient as menopause comes online. But all of these people were in the normal memory function, whether it was the associative memory or whether it was verbal learning and memory with the Bush case, which was really interesting. Now, I just want to say, way back, John Morrison has been studying the impact of estradiol on dorsolateral for many, many years. Those of you who are interested in this topic, John is brilliant, and I highly recommend reading Morrison. And he really found that an increase in estradiol, these were, he does monkey work, and he found in ovariectomized monkeys with vehicle, you could see that there was a dearth of dendritic spines. And when he added back estradiol, you have this beautiful spines occurring. And this was really in a similar region to what we were finding in looking at these working memory function. In this case, this was younger women. My former mentee, Emily Jacobs, who's now at the University California at Santa Barbara, did this in young women, and then we did it in older women, where estradiol was also really important in terms of neural efficiency. So again, just stating that estradiol is a powerful neuromodulator and impacts the changing of the brain in menopause. Okay, this is the verbal encoding test. I'll just say quickly, you have to form a sentence. You see these two words, and when you see a similar word after retrieval, you have to remember the sentence that you came up with. This is one version of it. We have several versions that we use. And what we found is actually a lowering of the left hippocampus only in postmenopausal women. Like women in general, premenopausal, perimenopausal actually did better than men in terms of activity of the left hippocampus. And actually, they actually had overall, women had better verbal memory function than the men. This is something that really, which I'll show you in a moment, really begins just post-puberty. But the postmenopausal women, even though they had similar memory function on cognitive tasks, actually were activating the hippocampus less. And what they did was they were recruiting the right hippocampus. So these postmenopausal women, to compensate, were recruiting the right hippocampus. And postmenopausal women who actually were high performers had less, let's just say there was less frequency of them recruiting the right hippocampus. But this was a compensatory mechanism that was important to maintain intact memory function in postmenopausal women. Okay, so now we want to relate, right? We want to say, well, now we see what's happening. We see what's happening from, this was 46 to 54. Can we actually look at maternal exposures early in development? Let me just say the preeclampsia fetal growth restriction, the reason we chose this among discordant sibling pairs is that it's related to a dysregulation of these immune markers specifically. And so we were really looking at variability of immune response and using obstetric complications as a model for that. And so now we're going to look at these immune markers in relation to their kids 50 years later. And what we see here is that this is maternal exposure. She's producing higher levels of IL-6 and TNF-alpha. And those that did affected a lower associative memory, particularly in the men. And when we looked at the sex difference, it looks like there was a sex difference. But there was a sex difference only with the pre and the perimenopausal women. The postmenopausal women looked like the men. And so the lesson for this is important. Many people, they look at sex effects and they just take their data later on and separate it by sex. This is not a way to look at sex differences, because if we had just looked at overall sex differences, we would think there's a difference, no problem, women doing better, blah, blah, blah. But it turns out that reproductive aging around this age is really important in driving various memory functions. So it's really important to think about where a woman is. Again, remember I said timing is everything. Where a woman is, even when they're premenopausal, within premenopausal, where women are even in their cycle. So we control for cycle, for example, for our premenopausal women. Okay. And finally, we can actually look at associations with the brain activity itself. So those men in which there were higher pro-inflammatory cytokines, TNF-alpha, IL-6, actually related to deficits in prefrontal cortex in the men. That was for working memory function. And then in that verbal encoding, we found that the connectivity of the right and the left hippocampus in the postmenopausal women actually related to a higher exposure to TNF-alpha in the mother 50 years earlier. So again, just making the point that actually we can map out there is something that gets retained into adulthood, and we can try to look at that, parse it out, think about it, think about how it affects men and women differently, and try to understand what are the processes that are driving this. Which, just leads me to the question of, why is it important to look across the lifespan? And that is, I'll just say quickly that sex differences in memory, in verbal memory, actually occur just post-puberty. So that there may be developmental roots to this, even in fetal development, but they actually express themselves just post-puberty when the brain is flooded with gonadal hormones in puberty. And that difference, this is just taken from a graph of a population study that started at 30, but you can see here across the board from 30 to 70, women have a higher verbal memory function than men. And even after menopause, there's an attenuation of the difference, but a little bit still gets retained in the healthy brain. So this is really important in thinking about memory decline later on and the risk for Alzheimer's and what might be resilient or not. Okay, why do we need to understand this? This is a slide I adapted from my colleague Lisa Sperling, also at Mass General. Really she's sort of looking at cognitive function and how it changes across the life. There's just normal aging, of course. There's the preclinical stage, and the preclinical stage has been identified at 45 to 65. Again, that's the period of the menopausal transition. So it's really important in this period to be identifying what are the sex effects in memory decline and how can we target this area for therapeutics that are sex dependent, right? We want to intervene early before we get here. So it's really important to think about these things and their developmental roots. That's going to get us to intervening early. One of the things we are doing is we are working on developing a clinical risk algorithm in early midlife that could identify who is it that's walking around with a lot of amyloid in their brain, but is cognitively, healthy, right? Those are the people we want to try to target for anti-amyloid drugs before they onset and before there's a lot of tau in the brain. And so we're very lucky that we have the Mass General Brigham Biobank. And this biobank has about over now 100,000 people in it. They're patients from Mass General and Brigham and Women's Hospital. And these patients have consented to giving a blood sample and having their electronic medical records connected to research and to the blood sample and to genotyping. And so this has been going on for several years, and those of us at Mass General or the Brigham can capitalize on this for different studies. So we are capitalizing on this by identifying a high and a low risk group that we're defining as having a polygenic risk score for Alzheimer's disease, which is been defined by Rudy Tanzi and his group, and or having one of these major chronic diseases that we're quite interested in, hypertension, major depression, or diabetes. And so that is our high risk group because, again, as I said, we're really interested in how cardiometabolic disease actually is a risk factor for AD and whether the sex differences in that can provide some insights into therapeutics, early therapeutics. So we first have to identify high and low risk people and look at all sorts of phenotyping. We're doing hormonal and immune. Obviously, there are a lot of genetics involved. We're doing metabolic profiling. And we're really interested in neurovascular structure and function. And we're doing very deep phenotyping. Initially, we have money for 100. We're submitting a proposal to actually carry this forward and increase the sample. But we're really looking to identify, using deep phenotyping, how to characterize these high and low risk people. And what we'd really like to do is come up with a set of criteria that a doctor could use in their office to identify who might be at high or who is at low risk for AD when they're still cognitively okay. And that's important without doing a lot of imaging. I mean, obviously, we're doing a lot of imaging to validate what the clinical phenotyping is, and we're using PET amyloid imaging to do that. But this study is moving forward. We call it the Healthy Aging Translational Cohort, because although we're starting with 100, we hope to actually create a much larger cohort out of this to really study all sorts of forms of aging and the sex differences in that. One of our goals is actually to try to create tools and therapeutics that incorporate sex differences knowledge into the development of the therapeutics. So the clinical risk algorithm is one, and this is another one we're hoping to create as a sex-dependent therapeutic called respiratory gated auricular vagus nerve stimulation. This was respiratory gated. Many of you may know other vagus nerve stimulation devices that are transcutaneous. They're transcutaneous, meaning that you stimulate a point. Actually, you stimulate a point in the ear, which is actually an acupuncture point. I'm pointing to my left ear. You stimulate this acupuncture point in the left ear, which actually is connected to the auricular branch of the vagus nerve, which is the primary afferent branch of the vagus nerve. And by doing that, we have activated the brain circuitry I showed you. So it gets at the stress circuitry and the vagus nerve. And we've shown we can activate that circuitry like the NTS and the regions in the cerebrum that the NTS is connected to. We have done, Vitaly Napadow really created the respiratory gated version of this. He's an engineer as well as a neuroscientist, and he studies pain. And Ronald Garcia and I have been applying this to major depression and hypertension along with Vitaly. And so we have looked at this in a number of instances where we've shown we can lower anxiety, we can lower depression through the stimulation, a certain, obviously there's a certain number of times over the course, but we really see some nice acute responses in people who are undergoing an episode of major depression. And we've also shown that it can raise cardiac tone and lower blood pressure. So we're really interested in looking at this also as a potential therapeutic for hypertension. And finally, I just want to say, those of you who are interested in memory, since the circuitry also really is shared with memory circuitry, we're quite interested in eventually targeting in early midlife memory function and mood. Okay, so that's, it's really important to us that we kind of reconceptualize precision medicine to incorporate knowledge, as I said, about sex differences in organ systems. Obviously, if there is a shared cause between depression and cardiovascular disease, and this occurs early, we could be targeting cardiovascular disease causes that are shared with depression at the time of post-puberty when sex differences in depression emerge, for example. Or if we were targeting cardiovascular disease, of course, if there are shared causes between the three of these diseases and we were targeting cardiovascular disease in someone's 40s or early 50s, we could also be preventing Alzheimer's disease. So really looking at this lifespan perspective, thinking about the sex-dependent aspects of these causal pathways are really important to the development of therapeutics, and we believe to really reach what everybody's talking about in terms of precision medicine. In order to do that, over the last couple of years, I launched a new center at Mass General Hospital called the Innovation Center on Sex Differences in Medicine. We call ourselves ICON. Our tagline is, when it comes to health, sex matters. And excuse me, this is a collaboration between Mass General and the Harvard T.H. Chan School of Public Health. we're also partnering with Women Against Alzheimer's, who's a policy and advocacy group for Alzheimer's specifically, but really interested in brain health in general. And so this is a collaboration where we are focusing on initially really on disorders of the heart and brain and the comorbidity of that. So we're really focusing on these three disorders that we talked about and trying to target in a therapeutic way comorbidity and the sex differences within that. And so I think I will stop there. These kinds of studies, when you follow large cohorts, they really take a village. I have an amazing team, but also we collaborate with many people across the hospital we're in psychiatry, but in neurology and OBGYN, in medicine, in cardiology. We have an amazing women's heart health program there that Melissa Wood heads up. So we are really lucky at Mass General that there's been a lot of engagement in thinking about sex differences in disease that cross organ systems. And these are some of my amazing colleagues. I also mentioned Stu and Bob Honda at Colorado State. And we also currently work with Stephen Gilman at the National Institute of Health and Development. I do want to say that I'm very thankful for being funded for so many years. The National Institute of Mental Health, Aging, NHLBI, and the initial work on Hatch is being funded by the Bright Focus Foundation, and I'm always grateful to our support from the Office for Research on Women's Health that supports a specialized center of research on sex differences at Mass General. Thank you.
Wonderful. Thank you very much, Dr. Goldstein. Wonderful presentation. So we'll go ahead and I'll check the chats. Right now we don't have any comments here, but I'll open it up. Does anybody have any questions, any comments for Dr. Goldstein? Okay, so we'll open it up to questions. Anybody want to, and if you want to unmute, or if you'd like, leave your comment in the chat and I can read it if you prefer. Well, I'll start with a question. I have a lot of questions, but I'll start with a question. I found it very interesting. You mentioned about glutamate and GABA in the hypothalamus, and you went quickly through that slide, but I'm wondering if you could, I mean, that's a very important question, and I'm glad you looked at, that level of detail. Based on what your research and what you've read, what are these gonadal hormones doing to that? I think you talked about the ratio or the balance between GABA and glutamate in the hypothalamus and how that's implicated in mental health and in your models?
Well, that's a great question, because what we did in those models were actually looking at the development of neurons into the paraventricular nucleus and into, well, it was really specifically that when Stu knocked out the GABA B receptor. We're interested in what is the role of GABA, for example, in major depression and in the emergence of sex differences in depression. That's sort of in the background, and we're actually doing a study on this now using spectroscopy. We didn't really do that in these studies with our prenatal cohort yet. So I'm not sure, from a data point of view, I can talk to you more about the fact that gonadal hormones actually receptors sit right next to GABA and glutamate receptors and can regulate their release. So there's a real crosstalk that that's true for every neurotransmitter, and that there are lots of differences in the metabolism of GABA drugs, and this is also, by the way, true for dopamine. Estradiol is a really important regulator of dopamine as well. We didn't really talk about reward circuitry or even the blood. I mean, the estradiol, for example, receptors sit right next to dopamine receptors and regulate blood flow. So they're really powerful modulators of lots of our physiology. And so those are some of the things we're very interested in looking at, but we haven't done a deep dive in the animal work yet to do that, except for, what I mentioned to you. Does that answer your question? We are working with someone who is a spectroscopist now to look at the role of the emergence of sex differences and the dysregulation of GABA. but that the jury is out on that one. But there is a basic biology to sex differences in the regulation of GABA
Okay.
and glutamate, but particularly GABA, actually. Did I answer your question?
Yeah, no, thank you. Okay. Do we have any other questions,
You know,
comments?
I will say these things are really important because, you know, there's only a few drugs that, as we say, are sex dependent. That really hasn't happened. But, you know, the GABA story is a really interesting one because I don't know whether you read about it down in Texas, but one of the Kennedys had a car accident. And so this, of course, made the news in Massachusetts. And so what they found were high levels of Ambien, which she had taken the night before. And what this sort of led to was a whole look at the clearance rates and also the impact of dose of Ambien in men and women, which apparently had not been publicized or put on, hadn't been marketed at all. And the FDA had to do a big, deep dive on this. And actually, it is one of these drugs now that is sort of dose dependent by sex, because they had even found data that they actually showed a sex difference in Ambien, in response to Ambien, but they didn't do anything about it because, you know, hey, it was just a sex difference. You know, that was years ago. But I mean, I'm saying that facetiously, but frankly, these things are not looked at. And I would bet that there are many companies with millions of dollars of drugs that have been canned, but the studies were not designed by sex. And so they didn't realize that maybe it was more effective in one sex than the other. This actually was true for a drug that they used for lung cancer that they were about to throw out, but found out that it actually had a major effect in women with passive smoking that's particularly high in women, and that the drug was unbelievably effective. It changed lung cancer in women treatment. But that was just a serendipitous finding by someone who actually thought about it. So this really needs to change. We need to be incorporating these things into the design of studies, not as a post hoc. So if you were interested in any interesting variable, right, you wouldn't just design your study, look at it, and later on kind of try to control away other things and look to see whether it was significant. Sex is complicated, and it needs to be designed into studies in order to find these effects, not as a post hoc afterthought. It's really important. I hope I've made that point. If anything, I hope I made that point to all of you who are, you know, doing work.
Okay, I have a comment. So what you find, and is completely has been found also in other fields, like in traumatic brain injury, where they find that was a glutamate excitotoxicity. So that go along with what you were saying, that the clearance of glutamate was slower in some condition of hormonal change in women, and there was an hypersensitivity due to that, and so that is very in correlation with your studies.
Very interesting. Oh.
Yeah, this is Murat. I am an exercise physiologist, so I'm looking things from a different angle. So one thing is the cardiovascular problems and those IL-6 exercise, all, and then right now, like this is interesting that I see that there are some correlations. By the way, I've missed the beginning of the talk. So again, if I am interpreting something or I'm misunderstanding something, so again, please forgive me. But so then again, with exercise, you actually have to release IL-6 in the system. And again, having IL-6 is kind of like training your body. And also you have to have this because for adaptation, you have to have those. And again, what I've been seeing is, again, if it's too little, that's not good. If it is too much, that's not good. But you have to have this, and we have to keep training our body, cardiovascular system. And I see that, again, IL-6, TNF-alpha, all of those are correlated with cardiovascular problems, brain, and but at the same time, again, I'm going back to this one because when you damage the muscle, you release IL-6 in it. And I don't know if you have looked at this specific IL-6. I know that there are some for heart muscles, there are some for skeletal muscles. But again, if IL-6 is in the system and it can come from the skeletal muscle, which is due to damage, and this is part of the training. And again, if you train, you get better cardiovascularly, you get better for again, in terms of skeletal muscle and neurological. But if it's too little, not good. Too much, not good. But I don't know how to, like, again, I wanted to see what you think about this, and then maybe comment on what I've seen, and what's your take on this one.
Yeah. So I think it's a really important point. I always think of this, it's like the three bears, right? I mean, it's like too little, too big, just right. And it's true of many different things, like pro-inflammatory cytokines are not bad. We need them. We need the response, right? But it's really true of many things. It's actually even true of an estradiol response. It's kind of interesting that you mentioned this kind of U-shaped curve, because for working memory function, it turns out that the comp gene, that your levels of estradiol will matter depending on the alleles you have in your comp gene, which is a estrogen metabolizing gene, but also really important in working memory. And colleagues found that if you have a certain allele, like a little bit of estradiol was great for memory, working memory function, but if you had a different allele, actually too much estradiol was actually not helpful. So these things are never black and white. I think that's an important point you made. As far as exercises, I think that's a really important and really bursting field right now, right? Sort of mapping out the biology of this. In psychiatry, what was found by some colleagues and others is that it actually enhances brain-derived nerve growth factor, specifically in the hippocampus, and which is actually also what was found with some antidepressants. And so it really has important effects on brain regions and releasing nerve growth factors as well in regions that are also regulating immune response, as you say. So there's a lot of crosstalk between that, and those regions, by the way, are sexually dimorphic, and no one's really looking at that from the brain perspective. So I hope you will, Murat, look at those sorts of things. I think it's really important, since BDNF levels actually produced by some of these brain regions actually have been found to differ by sex, but people are not breaking it down. They break it down by functioning and types of exercise and things like that, and looking at it in terms of risk for cardiovascular, but actually these things that are also regulating the brain are regulating the heart, and also involved in the physiology that regulates the vasculature and the neurovasculature. So it'd be really interesting if you have colleagues to work with you. I don't know if you're in neuroscience or whether you're a cardiology or cardiologist.
No, I'm like a purely exercise physiologist. So I'm actually, yeah. So
yes.
Again,
so,
by training,
my colleague, Judy Regensteiner, who's out in Colorado, who runs the Women's Health Research Center out there in Colorado at Anschutz Medical Center. She also does a lot of work in that arena, and sex differences in that.
What's the name again? Yeah, I have another friend working there too. So what was her name again?
Oh, Regensteiner.
Okay.
Regensteiner. Regensteiner. Judith.
Okay, thank you. I see.
she has a whole group there, I think, that do this kind of cardio. She's really interested in metabolic function, but she's got a group there that does a lot with exercise physiology. The other person who does a lot of work on sex differences is Mary, oh my God, Cohort, who's at the Mayo Clinic.
I see.
And you could look her up through, they have a SCORE, a Specialized Center for Research on Sex Differences. You might take a peek at that. They're doing some things with exercise as well. That's a sex difference center as well.
Awesome. Thank you very much.
Dr. Goldstein, thank you very much. This is Camille. I'm a psychiatry resident, and I'm actually working with Dr. Mario Gill here, who's here with us as well, on some project. And I was kind of fascinated by what you presented regarding the lifetime from the offsprings to the more than 65-year-old people. And I was wondering about the biomarkers that you were taking from these offsprings, and you said there's a collection of biomarkers as well that was taken. Is there kind of a list of these biomarkers, or kind of this cohort? Like, how can we know more about it, or how can we know more about this cohort so that we can get some data if that's possible?
So the National Collaborative Perinatal Project, NCPP, is actually public data.
Okay.
And that's all the maternal data in 1959, and it is a treasure trove. I mean, it is just amazing. In those days, they did such a deep dive into really finding out what was going on with the women during pregnancy. And collecting the blood, they have placental assessments of abnormalities in the placenta. It's very, there's a lot of, there's qualitative stuff and quantitative stuff. They did everything from eye and ear exams. I mean, we had a study many moons ago where we were really interested in kind of basic eye and ear abnormalities in primary visual and primary auditory cortex, as trying to pick this up as a risk factor for schizophrenia in early adulthood. And there was no one had ever looked at that because you wouldn't have the data pre the onset. And in fact, that is what we found. And it was unbelievable because it's mostly thought of as an information processing and not a primary perceptual problem. And so that was just unbelievable. Oh God, my postdoc never wrote that up. If you're interested.
Cool. And
I moved on to depression.
yes, anything. No,
but ours is, we also have a website, the New England Family Studies, or what our family studies are called. The website needs some help. You know, it's always the last thing that's on the list of to-dos, right? But you'll see some stuff that's there. And if it's not, you can just email me and ask me about it. But my colleague Steve Buca as Brown has sort of been heading up the sort of overall New England family studies. That some of them happen in the Providence area in Rhode Island, some happen in Boston. We do these things together. But Steve has kind of helped to put that, we've sort of put that website together, and he kind of is the holder of that now. So I'm not sure, I think it probably, the last time we spoke, which is, this is always a summer project, to upgrade it. But you really will have a lot of information there about the studies we've done and the variables that are in there, and things like that.
Thank you very much. I have just two very small questions about depression and the dorsolateral prefrontal cortex and menopause. Was that kind of taken into consideration? And also, if you have any comments about oxytocin and its role in kind of Alzheimer's prenatally versus like also as well?
Oh my God, that is so interesting. That I love reading about it. It's not my area of expertise, so I don't want to put my foot in. There's a huge sex difference, obviously, in the expression of that and so on. And you know, also a huge the olfactory system is highly sexually dimorphic as well, and that's sort of also been related to a lot of oxytocin related types of things. But I'm trying to remember the first question. Yes, depression. Yes. We are really interested in the mediating effect of depression, obviously. And in that study I showed you, depression can mediate and does mediate activity in a number of these regions. And we are really just working out the analyses. That first study ended just about a year or two ago, something like that. And then we were into following them again, and then COVID happened. And so we haven't gotten to writing that, but our preliminary work actually shows a mediating effect in some women of major depression on specific brain regions, as you said.
Okie dokes. Thank you. I'm done.
I can't remember about dorsolateral, but I can check that if you're particularly interested in that.
Thank you very much.
And we have a question from Dr. Angelica Potempa. Do you want to go ahead and ask your question, Angelica? No? Okay. Well, I'll ask it on behalf of Dr. Potempa. She texted it to me. So she talks about, liking the idea of these early interventions, right? So that was something you had mentioned with regard to vascular coronary disease. But she does have a concern. So she is a philosopher. So, and she mentioned, considering the problems of defining Alzheimer's disease, categorizing people at younger age at high risk could add stress, and possibly you can lead to over medicating. So if you could maybe comment on that.
Yes. First of all, yes. I agree. And I do think that that's a really critical point. It's really interesting that some people actually really do want to know their genetic complement and whether they've got the E4 allele or not. Others do not. There's a whole really ethical issues around genotyping young children. And just because a gene is associated with some of these disorders doesn't mean that it's going to be expressed. We know that. We know that none of these genetic complements, I mean, this is related to your question. I'm not trying to get around it. But it is true that having these things doesn't necessarily mean it's not deterministic, particularly around genetic complements, which we know have small effects, and in most complex disorders really have multiple small effects of common genes, let alone some major genes. They really haven't identified that, and you're right, with Alzheimer's in particular, we are not there for treatments yet. So what do you do if you identify it? Well, right now, Biogen is hopefully early next year coming out with an anti-amyloid drug that they think that they're showing is they have significant success with. Actually, it was in MCI. They haven't really used it in healthy people yet. But I do think that that's an important concern. I also think it's not necessarily Alzheimer's that you would be identifying. You can say that you are, like anything, you have risk factors in your family for X, Y, and Z. This clinical risk algorithm, you're right, needs to be named properly. And naming is really important. Naming and labeling is really important, can create a lot of anxiety. The same with genotyping with babies. What are you going to tell the parent, that this child has a gene for psychosis? We know that the genetic complements are not large for many of these single genes and so on. So this is I think this is a really important arena that needs to be associated with everything that we do, and particularly, you're right, with Alzheimer's, which can be frightening because of the lack of therapeutics. But I do think therapeutics are coming down the line. This Biogen drug, we're really crossing our fingers for success, and we'll see how much of the clearance there is of amyloid. But people actually die with a lot of amyloid in their brains, and they did not have Alzheimer's. It's a really tricky, disorder, but this is true for most complex brain disorders are tricky in their definition. And what produces this, and what's the combination of things that produce it, and what about the timing of talking about the risk for that? It's really important to think about those things. And you need a treatment, I think, before you're able to say we think you have X probability for getting a disorder like Alzheimer's disease.
I have a question. My name is Carlos Barrero from Temple University. Thank you so much, Dr. Goldstein, for this excellent presentation. I was very motivated. And I would like to hear what is your input related to the hormone replacement therapy, not only for like memory, like after the menopause, but also for like other kind of disorders like depression or all these things. Do you advise this will be beneficial? You already mentioned that it can change depending of the genetic background, but overall, like for the general population, what is your recommendation?
Yeah, it's a great question. Well, first of all, let me just say, I'm not a physician. And I'm not giving a prescription. The other thing is that, as you just said, Carlos, that these things are very individual. And so it's really important to know and talk with your doctor about what your history is, what your family history is as well, and to make these decisions. Okay, saying that, does estradiol affect memory function? Absolutely. And the question is when after menopause, or do you really target perimenopause? People are now thinking we should be targeting perimenopause. So the Women's Health Initiative, unfortunately, kind of put the kibosh on hormone treatment, which I think was extremely problematic. I was a colleague of Joanne Manson and still am, and she really felt that she re-looked at the data, and when she looked at the younger women who actually had therapeutics, hormone treatment, she found that actually it had beneficial effects. And it was really giving people hormone treatment 5 and 10 years after menopause. That is not a good thing to do. Your receptors will not be happy receptors ready to metabolize and deal with the estradiol. So you need to do it early, right? You need to make the decision early. And that decision might be during perimenopause, or it may be very early menopause. But the going sort of, discussion around this is, if you really wait beyond 5 years, beyond menopause, that's probably not great. You won't get the benefit, and actually there can be some deleterious effects. That's number one. Number two, so timing, again, is everything. And yes, it's been found that timing early actually can be quite beneficial. Now, if you have a history of ovarian cancer, or you've got the BRCA gene, or you've got the, whatever, these are other issues you need to take into consideration, although they found in terms of the breast cancers actually was more progesterone than a progesterone effect, even more than the estradiol effect. Okay, but the other thing that one needs to think about is the route of administration and also dose. So route of administration is extremely important, and also type of drug, type of hormone replacement. So are you using Prempro? I think, that's probably the WHI kind of put the kibosh on that. But what is the progesterone component? If you still have your uterus, what's the progesterone component in the HT? That's very important. The other thing is, now we have transdermal patches, which is a primary way that many women actually are on hormone replacement. That means it does not get metabolized through the liver, which actually can be quite advantageous for other organ systems. So transdermal, and really thinking about the formula of progesterone is really, really important for the health of other organ systems and timing.
Thank you so much.
I have, since you mentioned progesterone, a question that came to mind was, so the hormones associated with pregnancy, does that sort of change a woman's response to estrogen in the future? Have you looked at that, how hormones of pregnancy and things like that can impact the female brain and responses to stress and things like that?
Oh, that's a great question. I mean, hormones in pregnancy, oh my God, the brain is really, brain is bathed. And also, actually, I have 2 children. 3 months later, it's still bathed. It's really intense. But it can have incredible advantageous effects for certain disorders during pregnancy that have been found to attenuate. And it can have sometimes deleterious effects. So, and that's, for example, is one in which it gets attenuated, and lupus is one in which it might get exacerbated during pregnancy. But the brain does change with all this hormone stuff going on. How long that change occurs, I mean, you can see the representation when you have the immediate withdrawal postpartum with the onset of things like postpartum depression or other postpartum problems. And that's been attributed to this extreme withdrawal after pregnancy, sort of initiating a mood disorder for someone who may have the propensity for that. And so, that's just a clinical reality to that. The other thing are things like preeclampsia, again, which is a major risk factor for cardiovascular disease years later, even, after everything is resolved after pregnancy. Years later, it has a 3 to 5-fold risk of cardiovascular disease and serious cardiovascular disease. And then we also see even memory decline. And so targeting that postpartum period it could be very important for high-risk pregnancies. So those are the kinds of things that people have looked at. Whether that has an impact on the regulation of these things later in life and memory, there are disorders that have been found in women who don't have pregnancies. They have higher risk for certain disorders, and including disorders of the brain and memory issues. In part, that's thought because there is no relief of estradiol, cyclical estradiol during that period. So that has been associated, but have people actually done a lot of work in this area to try to mark out what are some of the mechanisms behind all of this? I hope you do it, Mario.
And I agree with the statement you made, especially the animal models and the biology. A lot of researchers just exclude females in many cases, or they don't really want to create a model that can translate to what women experience. So I think I hear you loud and clear.
And even Francis Collins heard us loud and clear in 2015. He required that that animals and cells actually be at least identified as male or female, and to design studies or to talk about why you're not designing your study to do that, which was a huge, huge step. I mean, that had a major impact on the academic community to do this. And sex is a biologic variable, the SABV policy for NIH now in the Office for Women's Health Research. This is pervasive now, and the Office for Research on Women's Health are putting out all sorts of courses for those of you who don't know how to design these studies. and examples of how to do this. They really want people to incorporate these things into everyday research. Still, most studies are male animals still. I mean, eating disorders have been based on male animals. Like,
Right.
unbelievable. It's unbelievable.
Okay, well,
And cardiovascular disease. And, cardiovascular disease is another really good example.
yeah.
And people were wondering why women with the same heart attacks were actually dying at a higher rate. They're getting into treatment later because their symptoms were not being recognized as symptoms of a heart attack. Mm-hmm. So even the symptom expression of a heart attack in women is actually different in men, and it turns out that it's a microvascular disease. And so, for example, I don't know if who is it who was doing the exercise?
He left.
Moreau was doing the exercise. The stress test for cardiovascular disease was built on men's health, and many times, for a large vessel disease, not for a microvascular disease. So many times a woman can get on a stress, do their stress test for cardiovascular disease and walk out and have a heart attack, which, unfortunately happened to a friend of mine.
Christine, you had a comment?
Yeah, about designing the studies for animals models with male and female. I agree with you, but the reason is that, for example, for the nervous system, because of the hormonal balance, when you have a repair of the nervous system, it depends on the hormones. And so then you will have a standard error too big and people to get results, they choose male.
Yeah.
Mm-hmm. So,
Yes,
so,
I know, and it's more costly. So, when Francis Collins makes the pronouncement, but then there's no funding behind it, that's a little problem. So I hear you, Christy, and that has been voiced. So what Janine has been doing, the head of ORWH has been doing, is trying to put out strategies for doing it that, sort of move us closer to tests of interaction effects, but not necessarily every study can do that. And you're absolutely right. You have to double your sample size, or at least double. You might even have to triple your sample size. Yeah.
Okay, well, it's 5:06. This has been a great discussion, wonderful talk. I want to thank our speakers. So a round of applause so you can clap, at home or use your reaction. So thank you very much, Dr. Goldstein. We really enjoyed the talk.
It was a pleasure to meet all of you. Thank you for having me.
Okay.
Thank you so
much.
much. Okay, bye.
Bye, everyone. Thank you.